Welcome to the second lecture in the second section in Advances in Microbial Pathogenesis and Biotechnology. This lecture will focus on diagnostic and control strategies. First of all, impact of new diagnostic method in the control of infectious diseases. We have a lot of new inventions and diagnostic methods that are significantly important in rapid diagnosis, and these save time, especially in major infectious diseases in the world. And we have this is because of a lot of limitation in relation to traditional diagnostic method because they are not rapid, they don't usually save time, they have low sensitivity and slow turnaround or turnaround time. The emergence of advanced diagnostic technology is serving to ensure that there is high sensitivity in diagnostic method, saving time and ensure that result is available within short period of time. The types of new diagnostic method include PCR, which is well known, such as direct PCR, PQC PCR, and a lot of other PCR. But more advanced and sophisticated methods are the CRISPR-Cas method, or the CRISPR in short, standing for clustered, regularly interspaced, short palindromic repeat. These are methods that is highly sensitive, highly specific, though is not time efficient because it's a method that involves genetic engineering, modifying viruses and bacteria, encoding genes to make sure that even the most systemic and inheritable diseases are cured. If you take example cases of diabetic mellitus, which type 1, as you know, people can inherit them from their parent. Now, with the use of the CRISPR-Cas technology, the genetic makeup of an embryo can be reached out. The section of the DNA that encoded insufficient insulin or lack of production of insulin can be rearranged and checked again, then replaced with normal set of DNA, which eventually the child can be delivered without any case of type 1 diabetic mellitus. So this is highly sophisticated method and it is well known in advanced countries and well used. Other rapid diagnosis techs which we are usually using here are the antigen antibody reaction. We use them in identification of different types of blood grouping, in rapid identification of HIV virus, hepatitis A virus, and the other serotypes, as well as also in the case of the identification of H. pylori, typhoid, and malaria. So they are highly specific by just antigen antibody reaction. So long as you have the antigen, when you cross the availability of the antibody in the human body, you can detect the presence of a particular pathogen. Then we have other methods that involve biosensorship and nanotechnology. We are using biosensorship to identify a lot of other biological markers as well. We are using nanotechnology in augmentation, synergy, and production of antibiotics, and also marking, most especially in the delivery of drug, in the mapping of tissue within and outside the human body, especially in histology, and a lot of other things. Now, in more advanced aspects of this diagnosis include the use of AI integrated diagnosis system, where the database will be uploaded with secondary data of a particular pathogen, and that can be used in the identification of pathogens. One example of this is the VETEC GNI system. a system where it is embedded with all the types of biochemical tests in the secondary data. So when a sample is run, the machine will compare it with the available data within the machine. Then an AI will interpret it to ensure that such kind of sequence of biochemical tests, they are compatible with the database. So technically, every bacteria that we know, it is embedded in the system, and whenever you run it, you can know which pathogen you have directly without going through the conventional biochemical method and phenotypic characterization. So you are bypassing macroscopy, microscopy, gram staining, and biochemical tests that we used to do. So this usually takes 6 hours, and you can have your result without any wasting time. Which recently, when you want to identify, characterize, take biochemical characterization and antibiotic pattern, it's going to take you 4 to 5 days. So now, using these advanced techniques, people can bypass the minute, the time, and we can get our result within a short period of time. Now, this diagnostic method, they have a lot of impact in disease control and identification. The impact is early detection. It is highly sensitive and specific. Therefore, detection can be done without even the manifestation of symptoms. And it is also used in surveillance and outbreak control. So, real time pathogen tracking. Real time without symptoms, whenever you have your sample, real time you will get your result. Example is the is the real time PCR. That is also used in the identification of variants and also outbreaks, where you can identify different biotypes and serotypes during outbreak. It also have impact in the antimicrobial stewardship. As I've said, the VTech GNI system is not only identifying microorganism, it is also testing some certain category of antibiotic against microorganisms. So you can identify an organism and also test it microbial profile to see whether if it is resistant or susceptible. So it reduces the time for the identification antibiotic pattern testing. As well, it slows the antimicrobial resistance because of the time frame we are using in the detection of the resistant pattern. Then it is also used in precision medicine, detection of resistant genes directly using, let's say, the multi-tap system, a system where pathogens directly can be embedded and all the available genes can be detected using the multi-tap system. And it is also efficient in host pathogen interaction properly, like in cluster formation, like in clandestine distribution and phylogenetic analysis. Then you can also personalize the treatment using a lot of other approaches. So the use of sophisticated diagnostic method is very efficient in the detection of microorganism and also sufficient in diagnosis of diseases. There are a lot of other underlying challenges related to the new diagnostic method. One is that the fact that they are very sophisticated, it means that they are highly expensive because most of them they use unique reagent, unique enzymes, and they need to be kept in a controlled environment where the weather can be controlled and the reagent as well can be controlled. And for you to operate them efficiently, you need the use of skilled personnel, which prior to the operation of the machines, a lot of other staffs that they are going to operate the machine, they have to be trained. Then there is limited access in low resource settings, especially in tropical Africa where the countries they are very poor and we have less sufficient and sophisticated public health system. There is limited access to such kind of machines. If I think we have only one VITEC system, two VITEC system in northeast Nigeria. I think we have one in Teaching Hospital Bauchi, and we have other ones in Yobe. So all these machines, like even the multi-depth system, is not, you cannot even find it here in Nigeria. Take example, the CRISPR-Cas method. No place in Nigeria can have such kind of system because of lack of other social amenities. Then the infrastructures were not even designed to house such kind of machines and system. Limited electricity, limited settings in the infrastructure, lack of and quick assessment to the reagents and the enzymes used in such kind of new diagnostic method. So there are a lot of other challenges bedeviling the establishment of such kind of diagnostic method. Now, one of the most prevalent and challenging infection in Nigeria is a malaria infection caused by Plasmodium species and transmitted by Anopheles mosquito. This is an established knowledge. However, the baseline is that you hardly find an individual in West Africa and Nigeria in particular without a malaria parasite. Whenever a malaria parasite is checked, as we mark it and we identified it, you hardly find an individual without plus one parasite. So technically, even people that were living in the presidential villa in Nigeria, they have a plus one level of malaria in their body. So that's why this is an infection of concern, and it is a re-emerging infection. A lot of money have been spent in prevention, treatment, and diagnosis of malaria. So it is a re-emerging infection of concern. So there are a lot of other parasites which they can cause malaria. Most prevalent that we know is if Plasmodium malariae, but we have oval, vivax, and also falciparum. But the most important aspect of in the life cycle of malaria is the fact that the vectors, they are always available. That's the Anopheles mosquito. They have been breeded everywhere. We have a lot of gutters. We have a lot of resting water in lakes and pond. These are their breeded environment, and the parasite is available. So the transfer of parasite is very efficient across individuals due to the availability of the vector. To make sure that we control such kind of re-emerging infection, we have a lot of multifactorial approach, and here is their overview, which is also similar to most of the evolving and emerging infections. For us to control them, first of all, we have to control the vectors. These vectors, they cut across a lot of other animals, and vectors control can be very straightforward. In some certain areas, it is recommended that we use larvicide or insecticide, as we normally call it, to control the larval stage of the vector if it is in a stagnant water like the fount and the lake. Where there is a high prevalence of the mosquitoes and other vectors, people are advised to use nets as well, then spraying the environment to make sure that people are kept in a very clean and hygienic environment. Then there is use of drugs and antibiotics. We have a lot of drugs that they are efficient in the treatment of malaria, typhoid, and other infections, but you know the baseline is that a lot of these drugs, they are resistant. So we are using such kind of antimalarial, antibacterial drugs to prevent the spread of such kind of infection. Now scientists are working over the globe to make sure that they invent or create a vaccine for this kind of re-emerging infection, most especially the malaria vaccine. The work is in the pipeline, and I think there are some test vaccines which we have been currently tested in a lot of other countries, but it's one of the most important factor. If we are able to make sure that we produce an effective vaccine for the prevention of malaria, this will be very effective in the continuous or total eradication of malaria. Then environmental management. This environmental management includes sanitation, making sure that all drainages are controlled without stagnant drainage, and also to checkmate urbanization to make sure that there are effective social amenities that can be used to contain the rapid spread of the vector and also the parasite together with the infection. And in the bottom line for every infection is people are not aware of the consequences, the mode of transmission, and a lot of other things. And that's why people have to be reoriented on the causes of malaria, the vector causing malaria, the malaria parasite itself, how it is disseminated, its mechanism of infectivity, and ways through which we can prevent ourselves from infection by these pathogen. So that's why community engagement is very effective and is very important in making sure that we have overcome the spread of malaria parasite and we curtail its prevalence in both rural and urban region. Now, as I've said, for us to control vector, we have to control the vector in its habitat, in stagnant water, in drainages as well. Then in our matrimonial homes or in our habitat through the use of net, example, insecticide treated net and long lasting insecticide treated nets. Then we can also use indoor residual suppression by using either larvicide or insecticide to kill the larval stage of the vector. As we know, the larval source of this vector is usually in stagnant water. That's why the use of larvicide can be effective in the prevention and eradication of the vector, or we can even use normal oil. When you cover the surface of the water, then you are choking the larva within the body of the water, and by extension, they are going to die entirely. Now, chemotherapy and prevention. We have a lot of combined therapy that we use in the treatment and prevention of malaria infection, and some of them they are very effective, but some they are not due to drug resistance. But so far, what we have is we have artemisinin-based combination therapy. We have intermittent prevention therapy and seasonal malaria chemotherapy, which some of them were being given to infant and newly born children. But we have a lot of other marketable drugs outside that they are effective in the treatment of malaria parasite. Now we have a lot of other vaccines in test. As I've said, some of them they are available now, some of them they are in the pipeline. So these vaccines, if they proven to be very effective based on genetic makeup in West Africa, because you know vaccines goes hand in hand with genetic makeup of the individuals. That's why I have variation of vaccines across so many continents and countries. So if they are effective in West Africa, I think we found a way through which such kind of parasite can be eradicated. So, and a clear prevention more than that is employment of the environmental management parameters through removal of stagnant water, which is very available now in Africa. Wherever you are, you see that you have gutter, which is a stagnant water, you have lake, you have pond. All these waters, they are stagnant and they are very good avenue for the breeding of the vectors and the parasite. And you know, in Africa, we don't have urban planning. There is no distance, no good ventilation, no road network in some certain streets, and this is also complicating the issue. And we are not very conversant, and we don't have efficiency or biological control methods. So all these are bedeviling the situation, and they are preventing the effective control of malaria parasite and other parasite alike. But the most important approach toward eradication of malaria as a re-emerging infection and other evolving infection, as I've said recently in the recent lecture, is that the One Health approach. A One Health approach is an approach that involves all host and all parasite, all microorganisms, a single approach that can tackle whole lot of infections in a one go. So that's why the One Health approach is one of the current research method that a lot of scientists are employing to make sure that we use them in the control of such kind of re-emerging and evolving pathogens. We can do it using integration of human, animal, and environmental health, and also sustainable disease control strategy, which is currently employed in a lot of other countries by the World Health Organization and the CDCs across all countries. And the major challenge in malaria control is drug resistance. That's why we have a lot in the market, and also there is a market control due to competition between companies, as well poverty, because poverty is playing a very important role. Companies are producing less expensive drug and less effective drugs, and this is also contributing to the resistant stewardship. Then the use of the insecticides. Now, because of the receded concentration of the insecticide, a lot of other larvicide and insecticide, there are also the pathogens and the vectors, they are resistant to this insecticide. Then we have issue of climate change as well. Then we have weak health system, which it is well established in Nigeria. A lot of the healthcare system is not effective. One bad thing is that you go to the hospital, you see that they are not even controlling their environment. A lot of herbs all over the place, a lot of stagnant water. There is no mosquito net, and there are a lot of other things where it's served the hospital, you can get a lot of other infections. So this is uncalled for and very unfortunate. So I think this is the end of this part. And when you finish, I think Dr. Umar Abubakar will take the third parts. And after you finish, before the commencement of the exam, we are going to arrange a CA test, which you as a PhD student have to be a very rigorous and envisaging assessment. So you have to look forward to it, and you have to follow this slide to make sure that you prepare a befitting note, which is peculiar to a PhD student. Nobody will give you a note, so you have to prepare it by yourself to make sure that you can cope with the current challenges. So thank you very much. I'm going to drop an MCQ question for you to follow and answer. This lecture will be uploaded in a particular server, then I'll give you the link to follow and download it for your own consumption. Thank you very much.